Hello class, in this video, we are going to talk about meiosis. This is actually just a continuation of our lecture this morning. Okay, so shall we begin? Good, okay. So, while mitosis consists of just a single round of cell division, meiosis consists actually of two. So, meiosis is divided into meiosis 1 and meiosis 2. But we are going to discuss meiosis 1 in greater detail because the events in meiosis 2 are actually just a repetition, almost just a repetition of mitosis. Ayan. Okay, so similar to mitosis, meiosis 1 is also composed of prophase 1, prometaphase 1, metaphase 1, anaphase 1, and telophase 1. But here, in meiosis 1, prophase 1 is divided into 5 stages. During leptotin, the duplicated chromosomes begin to condense, and they condense to the point that they become visible under the light microscope. Take note that this is leptotin. Technically, this is already part of meiosis, meaning to say DNA replication is already done at this point. Okay? So, again, ulitin ko, during leptotin, nagko-condense yung chromosome at dahil sa pagko-condense niya na iyon, nagiging visible na yung chromosome under the light microscope. The leptotin stage is, by the way, also known as leptonema. Then, the second stage of prophase 1 is called the zygote stage or zygonema. In this stage, nangyayari yung tinatawag natin na synapsis. Synapsis is the process kung saan nagkakaroon ng physical connections between the sister chromatid of one chromosome and the sister chromatid of the homologous chromosome. Pagtingin na lang yung picture sa PowerPoint, you can see there, let's say that long chromosome is chromosome 15, okay? Then, di ba sa assumption natin kanina, let's say that the blue chromosome is the maternal chromosome. So, yung blue chromosome na yan, na mahaba, that's chromosome 15 from the mother, tingnan nyo, yung sister chromatid niya ay nagkakaroon ng synaptonymal complex together with the sister chromatid from the chromosome 15 of the father, which is the red one. Do you follow? Okay, let's look closer. Let's say the dark band sa taas na part ng photomicrograph is the chromosome 15 from the father, let's say lang. And then yung lower edge niya, nandyan banda yung isang sister chromatid. Yung dark band sa taas, that's already two chromatids na magkadikit ha. So half of that dark band is one sister chromatid and then the other half is another sister chromatid. And then look at the dark band sa baba. Let's say that is the chromosome 15 of the mother. Again, consisting of already two sister chromatids. So yung upper half ng lower band is one chromatid and then the lower half of the lower band is another sister chromatid. So notice the lower band of the notice that the lower half of the upper band and then the upper half of the lower band ay magkatabi. And then in between them is mayroong whitish na region which is labeled in the photomicrograph as synaptonymal complex. So yung proseso na yan kung saan magkadikit yung dalawang sides ng dalawang chromosomes na homologous sa isa't isa. Again, when we say homologous, ibig sabihin same yung same yung numerical notation na naka-assign sa kanila. In this example, they are both chromosome 15, for example. So, ayan, yung proseso na yan kung saan magkadikit sila physically, we call that synapsis. And then yung aggregate of proteins na nagdidikit sa dalawang chromosomes na iyon, we call that the synaptonymal complex. Okay, so a little review. Again, in the leptonema or leptotin stage, nagko-condense yung mga chromosomes. And then in zygonema or zygote stage, nagkakaroon ng synapsis. May nabubuong synaptonymal complex sa pagitan ng mga homologous chromosomes. Okay? Take note na hindi nangyayari yung synapsis sa pagitan ng dalawang chromosomes na hindi homologous. For example, if chromosome 14 tapos chromosome 15, so they are not homologous, walang synaptonymal complex na nabubuo sa pagitan nila. The synaptonymal complex only forms in between two chromosomes that are homologous, one from the father and one from the mother. I hope you take note of that. So the end product of zygonema are chromosomes that are completely bound to each other, yung mga homologous pair, they are now completely bound to each other from one telomere to the other telomere. So magkadikit na yung mga chromosomes from end to end. Dito naman sa pachynemma or pachytene stage, ang unique na event dito is crossing over. In crossing over, yung dalawang arms, yung arms ng isang chromosome and then yung arm ng kabilang chromosome, yung homologous partner niya, ay nagko-cross sila sa isa't isa and then nag-exchange sila ng segments. This is an illustration from Russell to illustrate better how crossing over happens. Again, let us assume that the red chromosome is the paternal chromosome and then the blue chromosome is the maternal chromosome. Notice that both the maternal and the paternal chromosomes at this point are composed of two sister chromatids each. Now, yung right na sister chromatid ng paternal chromosome ay nag-crossover sa left sister chromatid ng maternal chromosome. As a result of crossing over, the terminal part of the right sister chromatid of the paternal chromosome now belongs to the maternal chromosome. It now belongs to the left sister chromatid of the maternal chromosome. Similarly, the terminal part of the left sister chromatid of the maternal chromosome now belongs to the right chromatid of the paternal chromosome. So yun, ang point lang talaga dito is nag-exchange sila ng segments. The region of the chromosome kung saan nangyari yung crossing over is called the chiasma. Now, take note that this process, crossing over, happens only in meiosis, specifically meiosis 1. We did not talk about crossing over in mitosis kasi wala namang crossing over in mitosis. This process class, the crossing over, is actually one of the reasons why meiosis 1 or meiosis in general is able to give rise to a very large diversity of combinations of genes and so a very large diversity of gametes. Meiosis kasi is that the type of cell division that is behind the process of gametogenesis. Kaninang morning, during our face-to-face discussion, I mentioned that there are two types of reproduction, sexual and asexual. I also mentioned that in asexual reproduction, which happens only by way of mitosis, yung daughter cells na napoproduce are genetically identical to each other, and they are also genetically identical to their mother cell. But in sexual reproduction, the two daughter cells are not genetically identical to each other, and they are also not genetically identical to their mother cell. So one of the reasons behind that is itong crossing over na ito. The other reason is yung tinatawag natin na independent assortment, which we will also talk about later. Now, after the homologous chromosomes have exchanged segments with each other, the synaptonymal complex will start to disintegrate. So, magsisimula ng maghiwalay yung mga homologous chromosomes. And that is what happens during diplonema or the diplotin stage. Finally, during diakinesis, the synaptonymal complex has completely disintegrated. So ayan, maghihiwalay na talaga yung dalawang chromosome, although sa illustration kayo medyo magkadikit pa sila gamay. But yun, the point there is nagdi-disintegrate na yung synaptonymal complex. And also, the nuclear membrane also disintegrates. Again, class, importante na mag-disintegrate yung nuclear membrane because the kinetochore microtubules have to gain access to the kinetochores of the chromosomes. Okay, so that's all for prophase 1. But before we proceed to pro-metaphase 1, nakalimutan kong i-mention kanina, so ngayon ko pa siya i-mention. Diba, during pakinema, tapos na or kompleto na yung pagkakabuo ng synapses or ng synaptonymal complex. Again, the chromosomes are already physically attached to each other from one end to the other end. The chromosomes become so attached to each other that the entire complex, yung dalawang homologous chromosomes na yun, we call the structure the bivalent, also known as the tetrad. Tinawag siyang bivalent because it is composed of two or dalawang chromosomes. Tinatawag naman siyang tetrad because it is composed of 4 sister chromatids. Kasi 'di ba, Each chromosome is composed of two sister chromatids at this point. So 2 times 2 equals 4. Kaya tinawag na tetrad. Again, bivalent, also known as tetrad. Now, during prometaphase 1, ay ang nangyayari dito is the kinetochore microtubules will attach to the kinetochores. So medyo similar lang siya sa nangyayari during prometaphase of mitosis, but there is one important difference. During the prometaphase of mitosis kasi, ang isang chromosome ay naa-attach siya sa dalawang kinetochore microtubule. One kinetochore microtubule from one pole, and then the other is a kinetochore microtubule from the opposite pole. So, ayun ha, again, in prometaphase of mitosis, dalawang kinetochore microtubules ang nag-a-attach sa kinetochore ng isang chromosome. But here, in the prometaphase 1, or the prometaphase of meiosis 1, ang kinetochore ng isang chromosome ay nakaka-receive or nakaka-attach siya sa isang kinetochore microtubule only. Nakaka-attach siya sa isang kinetochore microtubule coming from one pole. Please take note the differences between meiosis 1 and mitosis. Next is metaphase 1 or the metaphase of meiosis 1. In this stage, ang nangyayari naman is the chromosomes will arrange themselves into two files or two layers. Kanina kasi, di ba, sa prometaphase 1, yes, naka-attach na yung kinetochore microtubules sa kanilang respective kinetochores. However, the chromosomes are not yet organized. Medyo ano pa sila, medyo randomly positioned pa sila. But here in metaphase 1, naka-organize na sila, naka-arrange na sila along the metaphase plate in two layers or two files. Ang proseso ng metaphase 1 is actually similar lang din sa metaphase ng mitosis, although there is one important difference that you should take note. Yung metaphase kasi ng mitosis, the one on the right side of the PowerPoint slide, sa metaphase ng mitosis kasi, class, yung mga chromosomes ay ina-arrange nila ang kanilang mga sarili along the equatorial or metaphase plate in only one layer. Because the purpose of mitosis naman kasi is to divide or to separate the sister chromatids of the chromosomes. But here, in metaphase 1, the chromosomes are arranged in two layers because the purpose of meiosis is to separate the homologous pair. Okay, class. Ulitin ko, class, para mas klaro siya. The purpose of meiosis 1 is to separate the members of the homologous pair, to get one member of the pair and allocate that to one daughter cell, and the other member of the homologous pair and allocate that to the other daughter cell. So that's the reason why in metaphase 1 the chromosomes are arranged in two files. But in metaphase of mitosis, ang purpose kasi ng mitosis is to separate the sister chromatids of each chromosome, and then to allocate one sister chromatid to one daughter cell and the other sister chromatid to the other daughter cell. That's the reason why isang file lang yung arrangement ng chromosomes during the metaphase of mitosis. Now, I would also like to emphasize that the arrangement of the chromosomes into the two files during metaphase 2 is random. Ibig sabihin, okay, look at the illustration. So the cell is divided into two poles, or the cell has two poles. One upper pole and one lower pole, di ba? So, notice na yung yung dalawang red chromosomes ay nasa taas na layer, and then yung isang red chromosome naman, yung pangalawa, yung nasa gitna, ay nasa lower layer siya. That phenomenon, class, is what we call the independent assortment, which is isa sa mga principles of Gregor Mendel. Ganito lang naman talaga yung point ng independent assortment, class. Let's say you have chromosome 1, one from the mother, diba, and one from the father. So the destination of the chromosome 1 from the mother does not have an effect or does not have anything to do with the destination of the chromosome 2 from the mother also and the chromosome 3 from the mother. Okay, look at the illustration on the PowerPoint, yung nasa left banda. Let us say that the first pair of chromosomes na nasa pinaka-left talaga banda, that is, these are your chromosomes 1. Then the second pair, yung nag-crossover, these are your chromosomes 2. Then the third pair, chromosome 3. So, notice, class, na yung red chromosome 1 ay nasa taas na layer, yung red chromosome 3 ay nasa taas din na layer, but your red chromosome 2 ay nasa baba na layer. Similarly, the blue chromosome 1 and blue chromosome 2 ay nasa baba na layer. Then the blue chromosome, similarly the blue chromosome 1 and the blue chromosome 3 ay nasa baba na layer. Then the blue chromosome 2 ay nasa taas na layer. So what happens here is the chromosomes are not segregated according to sex. It does not follow na lahat ng chromosomes ay it does not follow na lahat ng maternal chromosomes ay mapupunta sa isang daughter cell, then lahat ng paternal chromosome ay mapupunta sa kabilang daughter cell. It's not like that. Instead, each daughter cells produced in meiosis 1, pag nag-segregate na ang mga chromosomes na ito during anaphase 1, ay yung chromosomes na marireceive ng isang daughter cell is a mixture of the maternal chromosomes and the paternal chromosomes. Do you follow? Now, during anaphase 1, ang nangyayari dito is similar lang din sa anaphase ng mitosis, kung saan the kinetochore microtubules will pull the chromosomes towards the opposite poles of the cell. The difference lang talaga is during the anaphase of mitosis, yung hinihila ng bawat kinetochore microtubule is isang sister chromatid. 'Di ba? Remember from the anaphase of mitosis, yung hinihila nila is isang sister chromatid lang, which is already equivalent to one chromosome. But here, in anaphase 1, the anaphase of meiosis 1, ang hinihila ng bawat kinetochore microtubule is dalawang sister chromatid constituting one chromosome. Sa lain pa nga pagkasulti, ang hinihila ng kinetochore microtubule during anaphase 1 is dyad. Remember the terms dyad and monad? Diba, when you say dyad, that is the chromosome composed of two sister chromatids. Pag sinabi mo namang monad, that is one chromosome composed of only one chromatid. So in the anaphase 1, ang hinihila ng kinetochore microtubule ay dyads. In the anaphase of mitosis, ang hinihila ng kinetochore microtubule ay monad. Ulit-ulit para mas mag-get. The mechanism that happens during anaphase 1 ay similar lang din sa anaphase ng mitosis. Remember yung napag-usapan natin kanina, the pulling and the walking mechanism na nangyayari during mitosis, similar process lang din dito sa meiosis. And then finally, nandito na tayo ngayon sa telophase. During telophase, na-segregate na natin yung mga chromosomes towards the opposite poles of the cell. And then telophase is also followed by cytokinesis, kung saan nagdi-divide din yung cytoplasm. However, please take note class na cytokinesis does not always happen in all cells in all species. So, parang optional lang itong cytokinesis na ito. Meron kasi mga cell class na nagse-separate yung chromosomes during telophase and nagre-reform din naman yung nuclear membrane, ibig sabihin nabubuo ulit yung nucleus, but the cytoplasm is hindi sila nagse-separate completely. Mayroon pa ring connection between the two daughter cells. So technically, hindi pa sila talagang separate na cells. For example, class, mas mapag-uusapan natin to sa ating future discussion, but ako na lang siyang ichika as early as now. The process of sperm cell production, we call that spermatogenesis. Okay, look at this illustration from Tortora, your favorite book for anatomy and physiology. So, notice na pangita ang label class na secondary spermatocytes after meiosis 1. Notice that the secondary spermatocytes, the two daughter cells produced from the primary spermatocyte, are not completely detached from one another. Instead, they have a structure called cytoplasmic bridge, which is you just understand the name as it is. It is a bridge composed of the cytoplasm. It is a bridge that connects the two daughter cells, which are the secondary spermatocytes. And then, after meiosis 2, after each secondary spermatocyte have produced their own daughter cells naman, which are the spermatids are also attached to one another, still by what we call the cytoplasmic bridge. So, my point here is, after meiosis 1, and even after meiosis 2 pa nga, minsan, hindi talaga nagse-separate completely yung dalawang daughter cells. In fact, in some species, right after telophase without the cytokinesis, nagpo-proceed na sila agad-agad to prophase 2. Now, before we proceed to meiosis 2, I would like to emphasize at this point that the product of meiosis 1 are two daughter cells that are not anymore genetically identical to each other, unlike the two daughter cells that are produced during mitosis. Bakit hindi sila genetically identical from each other? Number one, because of the crossing over that happens during what stage na yon ng prophase 1? Can you still remember? The crossing over happens during pakinema or the pakitin stage, okay? I hope naka-answer yung iba. Now, another reason kung bakit hindi na sila genetically identical sa isa't isa is yung process na na-mention ko kanina, which is independent assortment. Okay, look at the illustration again dyan sa telophase. So, medyo namumuo na yung dalawang daughter cell, medyo nagre-reform na naman yung nuclear membrane. Okay? Notice that yung namumuong daughter cell sa bandang itaas ay naka-receive siya ng dalawang red chromosome at naka-receive siya ng isang blue chromosome. And then, yung daughter cell naman na namumuo sa baba banda ay naka-receive siya ng dalawang blue chromosome and naka-receive din siya ng isang red chromosome. So again, let's say the red chromosome is yan yung paternal chromosome and then yung blue chromosome, that's the maternal chromosome. So you see the point there? Each daughter cell receives a mixture of maternal and paternal chromosomes, and that is independent assortment. Okay, at this point class, before we proceed, mag-segue sa takadali. Let us use a little of our imagination. Let's play with our imagination. So let us assume, class, na during prophase 1, walay nahitabo na synapsis, and then during metaphase 1 and anaphase 1, walay nahitabo na independent assortment. So what will happen actually is, since walay synapsis, then the maternal chromosomes and the paternal chromosomes will not exchange segments with each other. Therefore, they will remain intact. Maternal is maternal talaga na chromosome, then paternal is paternal talaga na chromosome. Walang halong mga segments. Now, since wala ding independent assortment, then each daughter cell will receive only either all maternal chromosomes or all paternal chromosome. Na-imagine ninyo? So if that's what happens, ang mahitabo is mura ra gid intaw nato og gi-recover ang sperm cell, ang genetic material ng sperm cell and ng oocyte na nag-form sa zygote, no? Kaya di ba, during fertilization, during the formation of the zygote, di ba na may duha ka cell na mag-contribute sa genetic material ng zygote, the sperm cell and the egg cell. So kung walay synapsis and walay independent assortment, basically we just segregated the maternal chromosomes from the paternal chromosome. So mura ra gid nimo gi-reconstitute ang duha ka gametes. But thankfully, because of synapsis during prophase 1, specifically during crossing over, happening during prophase 1, specifically during pakinema, and also the independent assortment during metaphase 1 and anaphase 1, because of those two processes, the two daughter cells are not genetically identical from each other. Yan. Okay, now let's proceed with metaphase 2. I'm telling you guys, metaphase 2 is just really very similar to mitosis. So ayun, during prophase 2, nagdi-disintegrate yung nuclear membrane na na-reform natin during telophase 1. And then during pro-metaphase 2, the kinetochore microtubules will again connect to the chromosomes via the kinetochore proteins. So similar na talaga siya sa mitosis class, because here in pro-metaphase 2, the kinetochore of each chromosome will connect with two kinetochore microtubules. If we contrast that to meiosis 1, to pro-metaphase 1, di ba, during pro-metaphase 1, the kinetochore of each chromosome connects to only one kinetochore microtubule. So dito naman sa pro-metaphase 2, the kinetochore of each chromosome connects to two kinetochore microtubules. So I hope you understand the comparisons. Please understand talaga yung comparisons, guys, because ito sila ang mga more likely na lalabas sa exams and sa quizzes. Now, yung metaphase 2 naman is very similar din talaga siya sa metaphase ng mitosis, kung saan the chromosomes will arrange themselves in a single file. O di ba? Mitosis kayang datingan. Isa lang ka-layer ang arrangement ng chromosomes here in metaphase 2. If you contrast that to metaphase 1, di ba sa metaphase 1, 2 layers ang gi-form ng chromosomes. And then, sa anaphase 2 naman, pareho ra gid pud kaayo siya sa anaphase ng mitosis. Di ba sa anaphase ng mitosis, the sister chromatids are separated from one another. They are pulled to the opposite poles of the cell. So pareho na po kayo diri sa anaphase 2 and different siya sa anaphase 1 because in anaphase 1 unsa man ang gi-pull towards the opposite poles of the cell? The dyads, di ba? Dyads. Katong chromosomes made up of two sister chromatids. Dito naman sa anaphase 2, similar to anaphase of mitosis, what we are pulling towards the opposite poles of the cell are not dyads but ano? Monads. Single sister chromatid lang talaga. And then, finally, during telophase, similar lang siya sa telophase ng mitosis and even sa telophase 1, similar lang siya. Ayun, nagre-reform yung plasma membrane to encapsulate the chromosomes that are now present sa opposite, sa yung paunti-unting nagpo-form ng mga daughter cells. And then, cytokinesis. And then you just review this table. This is basically the summary of the differences, the comparisons between meiosis 1, meiosis 2, and mitosis. So, kanya-kanyang review na lang. Now, the next concept that I will discuss is hindi ko siya nakita sa Brooker, sa Russell, sa Jersen, sa Lewis, and sa Tamarin. But since this concept kasi is very exciting to me, interesting talaga tong topic na to. I-discuss ko siya ngayon, although most likely hindi naman ito lalabas sa exam and neither in my quizzes, hindi din ito lalabas. Pero ichichika ko lang to kasi since mahilig man ako magchika. This concept kasi has something to do with the anaphase during mitosis and the anaphase 2. Kaya diba, during anaphase of mitosis and anaphase 2, what happens there is the sister chromatids are pulled apart. They are separated. Okay? The question is, how are the kinetochore microtubules able to pull apart the sister chromatids? Maliban sa walking and pulling mechanisms of the kinetochore microtubules, here is another answer to that. In histology, you learned that the organelle responsible for the degradation of cytosolic proteins is the proteasome. The question is, how does the proteasome that this particular protein has to be degraded? No, ginaunsa niya pag-decide nga kani siya nga protein is i-degrade or dili. Those proteins in the cytosol that are destined for degradation are actually labeled by another protein called ubiquitin. So those proteins that are to be degraded will undergo a process called polyubiquitination. Meaning to say, a series of ubiquitin molecules will be attached to that protein. Now, the proteasome will recognize those proteins na mayroong polyubiquitin na naka-attach sa kanila. Once na-recognize na sila ng proteasome, then the proteasome will allow that protein to enter the lumen of the proteasome and then doon na niya i-digest yung protein na iyon. Now, going back to our chromosomes, before anaphase, the sister chromatids of the chromosomes are held together by proteins called cohesin, from the word cohesion. So these cohesins are the ones that hold the sister chromatids together. So if you want to separate the sister chromatids during anaphase and anaphase 2, then we have to find a way to degrade these cohesins. So how do we do that? Sir, diraan na mosulod si proteasome? No, dili pa. The structure actually that degrades cohesin is not the proteasome, but the enzyme called separase. Okay? Separase. Tinawag siyang separase because it allows the separation of the sister chromatids. However, before anaphase, separase kasi is attached to another protein called securin. This protein naman is called securin because it secures the cohesion between the sister chromatids. When separase is attached to securin, separase is inactive. Okay? Pag sinabing inactive, ibig sabihin, hindi niya kayang i-digest si cohesin in the presence of securin. So in the anaphase, kung saan gusto nating nakadegrade si cohesin, we need to find a way to separate the separase from securin. Do you get that? Confusing ba ang mga words? Again, if we want to separate the sister chromatids, we need the cohesin to be degraded. And then cohesin is degraded by separase, which is which is inactive pa before anaphase. Inactive siya kasi nakadikit siya sa securin. So if we want to activate separase, we should degrade securin. The question is, how do we degrade securin? The answer to that is with the help of APC or C. APC there stands for anaphase promoting complex. Yung C naman, that stands for cyclosome. Tinawag siyang cyclosome kasi pinopromote niya yung progression ng cell cyclosome. Tinawag naman siyang APC or anaphase promoting complex because it promotes the progression of the mitosis from metaphase to anaphase. So anaphase promoting complex. So what APC does is siya yung magdidikit ng ubiquitin molecules sa securin. Do you follow? Di ba kanina na-establish na natin na those proteins that we want to degrade with the help of proteasome, kailangan muna silang i-label ng ubiquitin. And that is what APC does. It is APC that attaches ubiquitin molecules to securin so that proteasome will now digest securin, but it will not digest separase. So, ang result lang talaga dito is nase-separate na natin si separase from securin. And remember, separase alone without securin attached to it is what? Active. Si separase na walang nakadikit na securin, active na siya. Now, since active na si separase, kaya niya nang i-degrade si cohesin. Pag na-degrade niya si cohesin, then wala nang pipigil sa kinetochore microtubules sa kanilang paghila sa sister chromatids towards the opposite poles. Kahit kasi hilain ng kinetochore microtubules yung sister chromatids patungo sa opposite poles ng cell, hindi pa rin nila maseseparate yung sister chromatids as long as nandyan yung cohesin. So, yung goal lang talaga natin dito is to degrade cohesin. So, yun. Medyo complicated siya nga process, class, no? Don't worry, class. Again, this will not come out in the exam. Most likely. Kasi hindi naman, muna ko siya nakita sa inyong mga reference books. So, ako lang yung gichika karon because interesting siya na topic. Naamo ko din siya sa amo ang book dati sa Cytogen. And lahat na ang Cytogen books na ginagamit nato karon compared sa books na ginagamit namo dati. So, yun lang. That's all for meiosis. I hope na you learned something. You learned a lot. And see you sa ato ang next class. Bye-bye!